In this lecture, I will describe the Megatask crowdsourcing challenge for this course. The goal is to find relationships between changes in phosphorylation levels of cell signaling proteins and changes in gene expression in cancer cells treated with the same small molecules. The data that you are given for the challenge measures, for different cancer cell lines and small molecule perturbations, the effects on both transcripts and phosphosites. Hopefully, by using both types of data, we would be able to better characterize how cancer cells respond to perturbations by small molecules. Predictive models that can successfully guess the changes in phosphosite levels from transcriptomics could identify new relationships between short-term rewiring of the signal network to longer-term alterations in gene expression. The other lectures in this course cover the L1000 assay and transcriptomic data in general. So next, I will briefly describe the P100 phosphosite measurements. The P100 is an experimental platform which measures 96 phosphosites using mass spectrometry. The experiments are made in a high-throughput manner, enabling measurements across many different cell lines and small molecule perturbations. Similarly to the L1000 platform, the measurements can be extrapolated to a larger set of about 1200 phosphosites. Currently, there are 6 plates available with 5 cell lines and 46 small molecule perturbations. About half of these experiments can be matched to conditions in the L1000 gene expression dataset. To formulate the challenge, it's useful to have a little more abstract description. We imagine here that there are 2 multidimensional spaces of cellular states. One is the transcriptional state space, and the other the phosphoproteomics state space. At any time, a particular cell is represented as a point in each of these spaces. Here the blue color is one type of cell and the orange another type of cell. We now perturb the cells using small molecules, and the cells respond by moving to another state. The direction of change in the response to this perturbation we denote by the Greek letter delta. One could suspect that the direction of change depends on the original position. However, we hope that the change is somewhat independent of the origin. Then the prediction challenge becomes to predict the direction of change of the phosphoproteomic space from the direction of change in the transcriptomic space. Shown here are 91 unique and matched perturbation experiments. The direction of change are calculated using the characteristic direction method. The heat maps are visualization of the complete training data provided for the challenge. The data is available in the form of 2 CSV matrices, where each column matches 1-to-1. In summary, the challenge is to come up with a function that takes as input the processed L1000 gene expression data and returns as output the processed P100 phosphosite data. Models will be compared and evaluated based on the performance on future validation data. When submitting your algorithm, you need to provide a command line tool, the open source code, and a document describing your algorithm. Your submissions are due on December 3rd at 11:59 p.m. Eastern Standard Time at this website. You're free to work on this project as a team, and there are no grades associated with it. Good luck and have fun with the challenge.